So you have to block the virus without affecting the cells. And that's very difficult. It's different than parasites, bacteria, other organisms. Virus, finding a compound that blocks the virus, it does not kill the cells, is very difficult. So side effects are frequent. That's not acceptable. And each step of the viral life cycle involves host functions. So another difficulty is that some viruses of medical or veterinary importance, you cannot grow them in the lab, like norovirus, HPV, hepatitis B. You cannot cultivate the virus. How can you test an antiviral against the virus? Some viruses don't have animal models. You cannot test them in vivo. Only humans are the host. Some viruses are too dangerous. You need to have BSL-3, BSL-4 facilities. Very few places in the world have these facilities. We do have a BSL-3 at Fiocruz, like the SARS-CoV-2. You need to handle the virus under BSL-3 conditions. I will show the work that we have done with the SARS. So, another difficulty is that the compound, it must block the viral replication completely. Otherwise, resistance will arise. So, if you use different drugs of the compound, optimal dose, you decrease viral replication, viral titer. The intermediate dose, you decrease it, but mutants will be selected. And these mutants, they will be resistant to the antivirals. If you use low dose, that's going to be more intense. So no matter which antiviral you have, you are always going to have to deal with resistance. This is what viruses do, especially the RNA viruses. So it needs to target specific mechanisms of the virus, and this makes drug discovery expensive. So another problem is that most infections, I told you about these antivirals that are available against chronic viral infections. But most viral infections, like the ones that I mentioned, the arbovirus, they are acute infection. They are very fast, right? The symptoms last 1-2 weeks. You get sick. Rapid diagnostic tests, they are not readily available. So you go to the doctor. Sometimes you don't have the proper diagnosis because the doctor cannot clinically define which viral infection you have. You say, oh, you have a viral infection, right? There are no broad antivirals for treatment. And by the time you establish the treatment, the infection is over. So you need to detect it and treat it very quickly. Otherwise, the course of infection is very fast. So this showing, in this graph, how acute infection, how many days it lasts, and these are the chronic infection, the persistent viral infection. These are easier to develop antivirals. You have more opportunity for the antivirals to work. But for acute infection, it's very difficult. So safety problems, you should not be treating people, or you have a viral infection, I suspect you have fever, right? You have general symptoms. I'll give you antivirals. Except for the PrEP, the pre-exposure, for people that have HIV, that's going to have sex, right? It's recommended to have this pre-exposure treatment. But in general, you shouldn't be treating people without proper diagnosis. And as I mentioned, they are not broad-spectrum antiviral. So the path for antiviral discovery is a long path, and it's very difficult. If you start from 100,000 compounds, we need 100,000 compounds to get one final medicine, one final drug approved in the market. So it's 1 to 100,000. It takes years, 5, 10 years. So most compounds will be rejected because they don't have antiviral activity, or they are too toxic, or they fail in animals, or they fail in human tests. So you will start filtering these antivirals until you have a final molecule. The cost is also very significant. To develop one single antiviral, it's between 100 to 500 million US dollars. So only large corporations can develop, bring them to the market, because you need a lot of investment. It's very difficult. And these companies, they want to invest in diseases that affect the poor, because they cannot buy it, right? Like Zika, dengue, chikungunya, they affect the poor. That's why it's so important to have the participation of governments to invest it. Like in Brazil now, there is a clinical trial ongoing for dengue vaccine. The Butantan Institute, another institute in São Paulo, is conducting a phase 3 clinical trial. We may have very soon the phase 3 approved vaccine. They published the results this year, part of the result of the phase 3. The vaccine is giving very good protection. So we need a lot of investment. So now switching a little, I'll talk specifically about the work that we have been doing in our lab. So this is a work we did. The first are natural products that I'll talk to you. I am a veterinarian, I'm a virologist, so I do not understand chemistry. But we partner with chemists, right? They send us the molecules, either from natural sources or chemically synthesized, and we do the antiviral tests. So this was compounds that were isolated from the geopropolis of a Brazilian native bee. It's called the Jandaíra bee. So it's from the northeast of Brazil. So they collaborated to isolate it, prepared several fractions, and they gave us some compounds, the arginine and 7-methyl-naringenin. The structures are here, and then we tested them. First, this was my master's, now PhD student, Poliana. First, we tested the cytotoxicity. I'm not showing you all the details. And then we test the antivirals against Zika and dengue, and they have very good activity against both viruses. These are the data for dengue. We got similar results for Zika, which I not show you. So these are the fractions, the different extractions that they use. And then for antivirals, we always need to have the isolated compound. We cannot be working fractions. The journals will not accept the paper, right? So the collaborators that works with natural product wants to send the fraction, okay, but we need to have the isolated compound. So these are the fractions, and these are the isolated compounds. It does have a dose-dependent antiviral activity against dengue and Zika. So now Poliana is doing her PhD. These compounds that we studied, we are using nanotechnology to encapsulate this compound to improve the delivery. Because since we want to use it for Zika, it must cross the blood-brain barrier. So we have done all the nanotechnology characterization, and now we are doing the studies with the transfer to the blood-brain barrier, and then move into animals. So there's another work of Poliana. We got 2 papers with a plant from northeast Brazil. It's called Milona, Cisampelus ciampodalis. This plant, it had some reports from the Brazilian native people. We call them Indians, because in 1500s, Pedro Álvares Cabral was trying to come to India from Portugal. He was a navigator to buy spices. But the wind took him in another direction. He arrived on the coast of Brazil, in Bahia, and saw people very similar to us, the Indians and the Brazilians. He said, oh, I'm in India, and started calling them India. So we call the native Brazilians Indians. So the Indians, they use this plant to treat asthma. And it does have some activity against some respiratory pathogens. So this collaborator isolated the compounds. These are alkaloids, bis-benzyl-quinolone alkaloids, sent to us, and we test them, varifitein and methylvarifitein, against the dengue and against the Zika. We got 2 publications. This is another work that was done in collaboration with another professor, very good virologist in Brazil, Carol, since Women's Day, right? The PI, work in science. This in Brazil has become more and more popular. My lab has mostly women, right? So it's a Brazilian native snake. The snakes in Brazil are different from the rest of the world. So when the Portuguese arrive in Brazil, they will get snake bites and treat with anti-serum from cobra. It doesn't work. It has to be gender specific. In Brazil, there are 4 genders of snakes. So if people are bitten, you must have the anti-serum against that gender. Otherwise, it will not work. So this was discovered in 1900s. So this is a very dangerous snake, Jararacuçu. So Botrops. So these toxins of this snake, it has antiviral activity against Zika. We are doing a work with a Brazilian scorpion. It also has antiviral activity. So these are the things that people, especially the young students, they get very excited about rational pharmacal design, right? Rational drug design. I'll get the protein, I'll do docking, molecular dynamic simulation, right? That's very nice. But in most cases, antiviral discovery or drug discovery is by serendipity. You test the thing, right? It works. Sometimes it works in the computer, it doesn't work in real world. So, now I'm switching a little and talk about the synthetic compounds. Ronaldo, which is my friend, my collaborator that's here, is going to give a talk later. He has a library of compounds. And he sent us these compounds. These are triazole-based compounds. So this is a paper that we got against Zika. In my lab, we don't do high-throughput screening. We could do it, but we prefer to do the low-throughput screening. High-throughput screening, you get a viral protein, and you test different chemicals, right? Different drugs, and see if you can block a viral protein, usually a viral enzyme. But if it is blocking the enzyme, there is no guarantee that's going to block the virus, right? In my lab, we prefer to work with the real virus, right? The real thing. If it works, it works. I don't care if it's blocking the enzyme. Then later, we go to study the mechanism, how that molecule is blocking. Both approach, they have the pros and cons, right? Both, but I prefer to use the low throughput because we train students. We train the Brazilian virologists. In the Northeast Brazil, there are few virologists. Most of the universities are in the South and Southwest Brazil, São Paulo, Rio de Janeiro, Minas Gerais. So we need to train people to face the next epidemics and the next pandemics. That's why I prefer to use the low throughput. There's another work that we did, also using Ronaldo's molecules. These are different molecules. These are triazole pyrimidine derivatives. Ronaldo sent us 6 molecules, and this is a scientific initiation. In Brazil, we have a program for undergraduate students. We don't have at Fiocruz undergraduate degrees, but we receive them in the lab for internship. And they do like a small master's program. They stay 1 year in the lab, working 20 hours a week. They get a fellowship, so we have to have a project. They have to have a report. So these students, Melissa, she's 6 months in my lab. She's about to have a paper, a good paper. Because they get trained, when they get into master's and PhD, they are very well trained. When you go to the conference, they're going to present the results. So, she's working with chikungunya. And this is a plaque assay. It's my favorite assay in virology. Dubeco developed, right? Nobel Prize for it. Of chikungunya. Chikungunya is a virus that's very good to work in Brazil. I know that people from Europe here, I don't know what's the biosafety level of chikungunya in Europe. In Brazil, it used to be a BSL-3 virus. But since 2015, it's a BSL-2, so it's easier for us to work. We don't have to work under BSL-3 conditions, and the virus replicates very quickly. So these compounds, out of 6, these are the positive control, ribavirin. 2 of them have very good activity against chikungunya. We are continuing the studies. And now, to finish my talk, I will talk to you about the work that we have been doing with Dr. Ratti, British Ratti. Ronaldo met Ratti. He was revising one of Ronaldo's paper. And then Ratti decided to write Ronaldo. I'm going to write this Brazilian guy. And Ronaldo and I work very close together. That's how we met Ratti. And he sent to my lab some molecules. And we started testing them. And some of the molecules Ratti have done in his group some prediction. Oh, this one is going to block the 3C protease of the new coronavirus. Can you test it? We have a BSL-3 lab. So these are the several analogs. Ratti sent us hundreds of compounds. So the student is working a lot. So this is my student, PhD student, Renata. She works very hard, a very good student. Renata has, in her master, she finishes 9 papers, 9 high-impact papers. She has now, I think, 15 papers. So we have good students in Brazil. So out of these compounds, 3 compounds, 3G, 3E, and 4D, they have very good activities against the new coronavirus. I'm not going through the details, but they have very good activity, And Rati has done here in India some preclinical studies of these compounds, the safety profile. So these are the cells. We use there's several ways of doing it, the antiviral assay. In my lab, we use the CC20. That's here. The critical concentration for 20% of the cells. That means we get the drug, dilute it out, and we use the concentration that kills 20% of the cells. 80% of the cells will be healthy. That's the maximum concentration of a compound that we use. So if we get different compounds, we don't use the same concentration. Let's say 10 micromolar. No, we use the CC20 because we can compare it better. So people do it different. There are several ways of doing it. It's just your choice. That's our choice. This is CC20, half of the CC20, 1/4, 1/8. So we can calculate the IC50, right? The inhibitory concentration for 50% of the cells and the selective index. So they have very good activity against the new coronavirus. And this is an endless text. This is 7-MMPR, is a drug that when I got at Fiocruz, I'm a veterinarian. So we work using this drug for the canine distemper virus. There's a PhD student. And since I'm not a chemist, right, I don't understand well, but when I was revising his PhD thesis, he was about to defend, and Zika outbreak was going on. And I called him, Otávio, have you paid attention to the structure of this drug? Look at this. This is not a desoxyribose, it's a ribose. I teach molecular biology. So you have a hydroxyl here in carbon 3, and also have a hydroxyl in carbon 2, like RNA. So this drug had activity against canine distemper. It's a paramyxovirus, right? RNA virus. Tested against Zika, I think it's going to have Zika activity. So it does. We got a very nice paper. Said we always need to be doing research because the virus mutate. New virus arrive. So this antiviral discovery is an endless but very exciting task. So with that, I'd like to thank my group. So these are the people that do the work. I told you mostly women that get attracted to this lab-based work. Our collaborators, Ronaldo, Rati. This was Rati visiting our lab, 2019. I think we didn't get too much older, right? I think we are okay this year. And with Rati, the collaboration has been very productive. I was preparing this talk. I didn't realize this. But we got 7 papers with Rati in 3 years. And we just have one accepted. So we are very happy with this collaboration with Dr. Rati. So there's always opportunities for masters, PhD, postdoctoral. So be welcome to join us and visit us in Brazil. Thank you. Dhanyavad.
So paper is open for discussion. Maybe 2 questions.
Thank you for providing such grateful insights about this topic. But I would like to ask a question. Can immunomodulatory agents or immune checkpoint inhibitors be combined with antiviral drugs to enhance therapeutic efficacy or not?
Oh, that's a wonderful question. And in fact, we do have, there is an approved medicine that's used for treatment of hepatitis C that combines ribavirin, it's a chemical, with interferon alpha, pegylated interferon. So it's an immunomodulator and antiviral. So these 2 molecules work very nice. And sometimes also, we need to think about for controlling diseases. I always think on both sides, humans and veterinary, right? I work with foot and mouth disease in the US. If you have a vaccine against foot and mouth disease, it's a very acute disease. It's the most contagious disease known to mankind. So the R0 of COVID is 2, right? 1 person infects 2 persons. Foot and mouth disease is 80. Infected cow will infect 80. The safety distance of COVID-19 is 2 meters, right? Foot and mouth, 2 kilometers. The virus is so contagious and it's so fast. That's why you have to be vaccinating cattle all the time. Because if the immunity goes down, if you have an outbreak, the vaccine will not protect. So you have to have very high levels of neutralizing antibodies, which is the correlate of protection for foot and mouth. So what we did at the time, if you vaccinate the animals, they'll be protected by day 7. You get protection. But you have a 7 days window. For virus that's so contagious, that's too long, right? The disease spreads very quickly. So we combine the vaccine with interferon alpha, like a strategy similar to AstraZeneca, right? That was one lie. I don't know if this lie happened in India, but there's a right party president in Brazil, anti-vaccine, a denialist. He was against vaccines. And one of the arguments, they say, oh, the COVID vaccine was developed too fast. 1 year. How come? If you take 10 years, right? But that's a lie. In 2005, we will develop this vaccine for foot and mouth, very similar to the AstraZeneca, using adenoviruses to express the vaccine and also to express interferon alpha. With that combination, you get protection from day 1. So the interferon will protect in the first week, and then after the first week, the vaccine will protect the animals. So in veterinary medicine, the field of vaccinology is much more developed than human, because you can do the trials, right? Very easy. There is no, there are ethical concerns, but not as difficult as to doing studies in humans.
And so how do you assess the preclinical, for the activities, and how do you select patients?
So for this work, we are doing some animal studies. We do the preclinical work in animals using mouse models. So there are some difficulties for that. Let's say Zika. If you get Zika and try to infect the conventional mice, they will not develop the disease. They will have no signs, so how can you compare an animal that's been treated with a control? But we have developed a Zika strain, mouse adapted. And also can use, we do have in the lab the AG129. It's a knockout mice for interferon alpha and gamma, type 1 interferon, type 2. So these animals, they develop the disease. So they can be used for antiviral testing.
Thank you very much, sir.
I think the more question can be asked during the tea time, because time is short now.
Thank you.
Thank you very much. So our next
thank you, sir.
speaker is Dr. Rupesh Chaturvedi. Dr. Rupesh Chaturvedi is from GNU. I'm sure everybody knows GNU. Jawaharlal Nehru University. He works in the area of infection biology and including even cancer also. He has published large number of excellent papers. Yeah, so maybe I'm sure you will hear from the Dr. Rupesh Chaturvedi.
Thank you, sir, for introducing me, and thanks. Good morning. Thanks for coming to hear us. This is a very interesting session where you just learn about how the antivirus discovery could be a challenging. And I'm going to just discuss about a model which we are creating, which might help. Where there is not an animal model, but still you can use some non-animal human models to do the study. So as you can see here, it's mostly, it's what we are trying to do is spheroids. Spheroids are basically, it's a three-dimensional structure of any given a tissue. So we take cell lines or cells from animal or the human, and then we can grow a mini size of those organs, and I will show you how that looks like. But if you think about, just about a cancer, and you think about how the cancer is heterogeneous in nature. We got the idea of doing this just by looking at the literature where they talk about there is a genetic heterogeneity into the cancer. Question is, if there is a genetic heterogeneity in a cancer, can we capture it for the functional purpose? So this is what we saw, and then we started doing some work on that. And what we did, rather than, if you look at most of the spheroid literature, what they do, they use a large number of cells to grow these spheroid in some kind of a substrate or in a low attachment, ultra low attachment dishes. And what happens, they comes together and then they form a 3D dimensional structures. What we were interested to know, can we capture the functional heterogeneity? When I say a functional heterogeneity means, is there some clone which is proliferating faster or migrating faster, and there are some clones which are doing not so. So what we decided to do is, we decided to grow these spheroids from very low number of cells. And for this purpose, we used only 2 and 4 cells, as you can see here. And what we got remarkably is that there was a 2 different type of clones we observed. And there was, although there was a different frequency for generation of these bigger size and the smaller size of spheroids. And you can see here, you can see here that the cells which are hyperproliferative spheroids, they grow much faster compared to the hyperproliferative spheroid. Just to see that what are they of the functional in nature, you can see that we stained them with a calcineum, which is stained live cells, versus just a counter stain, and ETBR, which is a dead cells, and they look like they are functional in nature too. Just to show this better, you can see here that this is a hyperproliferative. You can see how nice structure is there. Then lot of cells which is dead, and this is going in a lumen, showing that this structure has a polarity, intact polarity. Otherwise, these cells will go outside. So that's what you can see here. This is just a confocal image for both hypo and hyperproliferative cells. And you can see here most of the cells which is in this movie, you can see that the cells which are dead is in the lumen, showing that whatever we are seeing is a functional in nature too. So then we decided to look at, is this is happening in a human also from a primary tissues, and we saw the almost same, we had the same observation. But one thing we saw that was very interesting, if you look at growth of hyper versus hypo, as this stage of cancer increases, we see a more small size of a spheroid compared to higher size of a spheroid. This is to show that there is a differential effect of anti-cancer drugs when we compared hypo and hyper spheroids. And you can see here, this is a hypo and this is a hyper. If you look at just the ETBR, and you will see that with the radiation or with the drug, there is a less death of cells compared to the spheroid, which are hyperproliferative in nature. And what this says to us, this says that there is a functional difference is there. So if you suppose that there is a cancer in a patient, and this is from the cancer patient spheroids. So if we are treating, if somebody is treating with, radiation or with a drug, these two have a two different level of sensitivity. And with the optimal dose which have been used, or this number, this amount has translated, probably these cells are not going to get killed, they will still survive. So what is happening when we were doing a chemotherapy, probably we are selecting for the survival of these type of clones, which later on can cause the recurrence or progression of a disease. So this is what we are proposing, that the system can be used for the purpose of a drug discovery as a platform. This is some data from xenograft model, and you can see here that when we inoculated the hyperproliferative spheroids in a nude mice, they were not able to initiate any cancer, whereas the 50% of with the hyperproliferative did initiate the cancer. And the most interesting part we saw is the survival curve. You can see here in the mice which were transplanted with hyperproliferative, they did not survive that much. And it seems like that there is something in these spheroids are, and we know that they are also hypermigratory in nature. So it could be possible the time we were putting it there, it's metastasizing to the other part, and these mice are dying. So just to find out what is happening, how these two are different, hypo and hyperproliferative spheroids, how genetically they are different. So what we did the single cell transcriptome analysis, and you can see here, this is A is your data from, this is it's a statistical way to show type of a different type of a cells into hyper and hyperproliferative spheroid, and you can see that there is a two different pattern in terms number of spheroids are. When we look at their So you can see here that, and these clusters are identified, there is a certain number of genes are there which identifies these clusters. Then next, we looked at, is there any difference in terms of how these clusters might be regulating each pathways, and then certainly they might be also regulating other clusters also. And you can see that there is, in a hyperproliferative, we see the Wnt pathway, VGFR pathway, and the proliferation migration pathway, which are getting enriched. And the same way here, and there are some very specific pathways we have noticed, like TGF beta pathways. So then we thought to look at, is there difference in terms of what is the cellular lineage is there, what is the cellular ontology is there, and you can see that they have a very different type. This is the hyperproliferative, and this is the hyperproliferative spheroids, and you can see that, remember, these spheroids are grown from only two cells, and they were epithelial in nature. And what we have observed, that you can look at that there are some mesenchymal and the fibroblast cells here. And other than that, this epithelial cell is also differentiating and terminally differentiating, and there is two different lineages we can see in a hyperproliferative versus hyperproliferative. So keeping this thing in mind, these spheroids, in their clonality, they are depicting what might be happening into the system. And this can be utilized for further drug discovery part. This is a cluster-based analysis we did to look at what is happening in terms of actual patients. This is the data fetched from TCGA, and from each cluster we had a 50 different type of genes, which we used to do the signature expression, so cumulative expression of a 50 type of different genes. And we found that in a hyperproliferative, cluster 3 is mimicking what we saw in a spheroid versus a patient, and whereas in the hyperproliferative cluster 1 was genes are the markers which are present into the patient, suggesting that this is there. And and it suggests that genetically and functionally they corroborate. There is a difference at the genetic level which is manifesting at the phenotypic level. And this is the reason why we are using it as a tool to do the drug discovery or for the diagnostic purpose. I will switch from here how we are utilizing this spheroid for using it as a tool for in infectious disease. So you all know that there is a bacteria called Helicobacter pylori. In India, most of the people, 80% people, are infected with the Helicobacter pylori. Due to some reason, we don't develop a disease. Few of us develop gastritis, and then it progresses to the cancer, which is a stomach cancer. So we thought that, okay, can we create a spheroids which can mimic the stomach, and then can we utilize to do the functional studies. This is a mouse gastroid or mouse gastric organoids, and you can see here, it's you can see here, this is just one layer of epithelial cells, pretty much organized in the nature. And this is just we did the mucin staining, and you can see that these are the cells, and this is an apical part of those junctions, and these mucins are expressing at the apical part, and just to show that they do the retrograde transport and they go into the lumen, suggesting that this is functionally intact stomach. So another group from our team, they showed that this is a more functional in a nature, and you can see here that these are the cells. If you look at the D, E, F, and G, they are different type of differentiated stomach epithelial cells. Like D is your mucin, E is parietal cells which makes the acid, and then this is a gastrin, and this is a chromograin producing cells out there. So we are creating these things from two cells, but they are differentiating pretty well. And now these can be used to do the the study how in a three-dimensionality or 3D space how your infection takes place. You can do a lot of imaging on that to show that how exactly these pathogens binds. It's whether H. pylori or you can utilize for any other diseases. So like you can see here, this is occludine in a control, and what we did, we infected with H. pylori, and these green strains, they are H. pylori, and they basically co-localized with the occludine, which is a tight junction protein. And you can see that otherwise, if we do at the 2D culture, sometimes it's very hard to really get this information out from there. So this is like a more functionally what happens into the stomach during the infection. This is just to show that that H. pylori, this is a one strain which causes a cancer, and if we knock out CAG-A, which is a virulence factor in a CAG-A, and if you look at how the proliferation is happening, you can easily show in here. H. pylori causes a proliferation into the into the gastric epithelial cells. Whenever we have done with the 2D culture, we are not able to see this BRDO incorporation. We have only seen when we switch to this spheroid and organoid model here. And this is the beta-catenin, showing that how the beta-catenin can translocate, can activate in the whole process. This is another a model where we showed that H. pylori, especially, this is every time this is happening. We showed that H. pylori causes activation of a CSAR, and then the curcumin, which is a phytochemicals, can inhibit this phosphorylation of a CSAR. And this is what it's a Western blot coming from here to show, we are showing it. And it's not only provide us an idea that phosphorylation is happening, but it provides us an idea where the phosphorylation is happening. Because these molecules, if they are getting phosphorylated at the apical surface, basal surface, or what type of the cells they are getting phosphorylated, will have a more profound effect deciding what type of therapeutic drugs can be used. This is the last slide I would like to show you, that when we infected H. pylori to a normal mice and we took out their gland and we made the spheroid, you can see here it's a nice structure. Is here, mucins are at the apical level in a both. And then what happens, this is the INS gas mice which develops a cancer with H. pylori infection. And you can see in the INS gas mice, what we see is the structure is inside out, and they are dysplastic in a nature. This is see how this gland is well organized, whereas this one is not well organized. And the difference is, other difference you can see here, mucin is the outer side. Means the whole organoid is inside out. So this is a one of mechanism, one of the methods one can utilize to do infection biology, or to utilize it for drug discovery purpose. This is my collaborators, my group collaborators, especially Vibha Tandon and Rana P. Singh. They were both professor in JNU, and then our clinical partner Hitesh Verma. Thank you. Thank you very much. And this work is funded by DPRP program. Thank you very much. If you have a question.
So one question, please, if anybody has.
Yeah. Can you say?
Hello.
Yeah.
So is it same like the 3D tissue model? These spheroid models are used for the infection, and we can study on them. So my question is, apart from the morphological structure, are the gene expression or the transcriptomics level are same with the mouse model? Although it is really good for us that it will reduce the dependency on the animals model. But my question is it same with the transcriptomics level also? Because
so you might have seen that that single cell transcriptome analysis, which is we have done it from the patient also, and we see that there is a pattern of clusters there, and those clusters are defined by some type of marked by some type of a gene expression. And when we took the gene expression and the cluster wise, we looked at the TCH data set, we did see a good correlation there. And I showed you as in the talk that especially cluster 1 and cluster 3 of a hypo and hyperproliferative, they correlate pretty well in the human studies when we did the analysis in the TCGA data set.
Thank you, sir.
Rupesh, I have a
Yeah,
small
sure.
question. In your hypo and hyperproliferative cells, chemo may be different, may give different results because the uptake could be higher in the highly proliferating cells.
Yeah.
But why radiation was different in both?
Yeah, so that's the thing. If you look at so we are working on that part, looking at how the radiation effect is different in hypo versus hyper. If you look at the hypo, in a hypo there is, when we did the pathway analysis, there is a DNA damage repair mechanism is upregulated at the level. They should die with the radiation, but this is what the difference we are seeing. But I think it's mostly the genetic makeup of the hypoproliferative, which is different than the hyper. In hyperproliferative, we see a enrichment of the DNA repair mechanism. So it could be possible those repairs are happening, so there is no need for going into the apoptosis.
Thank you very much. Principal scientist in the Division of Molecular Microbiology and Immunology at CDRI, Central Drug Research Institute, Lucknow. He did PhD also from CSIR lab, NBRI in Lucknow. For his postdoctoral part, he has worked in Johns Hopkins, he has worked in NYU, Weizmann Institute, so many places he has worked. And he has got several awards, which includes Venkatraman Memorial Best Scientist Award from National Academy of Biological Sciences, Dr. Tulsidas Award, Shakuntala and Amir Chand Prize from ICMR. So it's a great achievement, and request Dr. Mishra to start his talk.
Thank you, sir. So good morning, everyone. Thank you, organizer, for inviting me and giving me opportunity to talk about my work. Wonderful hospitality indeed. So today I will be talking about the multifunction of autophagy pathway in malaria parasite and how we can target this pathway to develop a drug that not only kills the blood stages but also kills the parasite in mosquito and also in the liver stages. So malaria parasite complete its life cycle in two hosts, mosquito vector and mammalian host. So when human is infected with the malaria parasite, so parasites are circulating in the blood. They go through the erythrocytic cycle, and during that cycle they produce the gametocytes. These gametocytes are when a mosquito probe for the blood meal, so they are taken up, and within the mosquito midgut they immediately converted into gametes. Those fuse to form the zygote, and these zygotes develop into the ookinets, and then it infect the midgut wall that form the oocyst, which forms thousands of sporozoites, and then these sporozoites travel to salivary gland, and when mosquito again probe for the blood meal, it inoculates sporozoite in the human skin, and then from skin they travel to liver, mature into thousands of merozoites, and then finally they reach in the blood. So during this whole transition process, one of the process is transition of banana shaped sporozoites into the round liver stage forms. So during this transition, some of the molecules and some of the organelles and the storage like micronemes, they are not required during liver stage development. So they get eliminated, and during this elimination process, parasite exocytose by using this autophagy machinery. So if you look at the structure of sporozoites here, so these contains these red ones, so these are micronemes. So when parasite enter the liver, it gets converted into the round form, and then these are exocytose, and then this autophagy pathway means ATG8 conjugation on the membrane is required for successful development of the parasite within the liver, like organelle biogenesis and formation of the merozoites. Okay, so this autophagy pathway involves, this is the canonical one, so involves the conjugation of ATG8 molecules when it initiates. So for this initiation, when its ATG8 is conjugated, so there is a formation of isolation membrane, and this isolation membrane finally form the autophagosome, and then that fuses with the lysosomes, and that degrades damaged proteins or maybe damaged organelles. But in case of Plasmodium, there are limited set of genes. If you look at the mammalian system, there are 43 autophagy genes compared to the Plasmodium that is having around 14 genes. So they have only non-canonical ones, and the only report which suggests that probably this is involved in the apicoplast biogenesis, not this canonical one like appearance of the phagosome membrane and fusion with the lysosomes. So this whole pathway involves ATG8 conjugation. So for this one, initially ATG8, where there is a one penultimate amino acid glycine, so that is required for the conjugation. So before this one, this is processed by a cysteine protease. So this cysteine protease process it, and then ATG7 is a activating enzyme. So after ATP hydrolysis, it activates and forms thioester bond with the cysteine, and then after the action of E2 and E3, these three enzymes, finally ATG8 is conjugated on the membrane. So this whole pathway requires function of ATG4 during the initial processing of ATG8, and when this ATG8 is conjugated on the membrane, then again it's a deconjugated. And the ATG7 is also involved in ATG8 and ATG12 conjugation system, because this acts as a activating enzyme in both the conjugation system. So we targeted both ATG4 and ATG7, and the first thing we checked whether ATG4 process the ATG8, because in case of Plasmodium parasite, the terminal glycine is already exposed. It's not masked by another amino acid. So what we did, we masked its terminal glycine by fusing it with the HA and M cherry. So by using this transgenic parasite, first thing we checked its expression and localization, and we found that it expresses in the cytosol and also on the apicoplast membrane, because it localizes with the apicoplast marker. And then we expressed ATG recombinant, and by using this recombinant protein, we showed that when we incubated with the cysteine protease inhibitor, there was no processing. However, in case of like without E64, we can see that this ATG8 M cherry fusion protein is processed into HAM cherry and ATG8. And we also showed this deconjugation by using the membrane fraction of the parasite. So for this one, we purified the membranes from the blood stages, then incubated with the recombinant ATG4 in presence and absence of cysteine protease inhibitor E64. And we clearly show here that in absence of cysteine protease inhibitor, there is a processing. However, when we put this inhibitor, there is no processing. So parasite possesses this activity of processing ATG8. And next, so by knowing the importance of this ATG4, we generated the knockout by using the double crossover homologous recombination, and we confirmed this knockout by integration of this GFP cassette, and the blood stage parasites are expressing GFP, and after knocking out, we found normal growth of the blood stage parasites in both knockout clones, suggesting that ATG4 might not be important, or maybe ATG4 is absolutely dispensable. So what's happening? नहीं यही दबा रहा हूँ. It's not working. Working? Okay. So next we check the role of ATG4 in deconjugation. Like when ATG8 is conjugated on the membrane, it should be deconjugated for the proper development of the apicoplast. So for this one, here we did the localization. So there is some issue. Can you fix it, please? wait. Okay. So we checked its expression and localization, where we saw that in knockout parasites there is a higher level of ATG8 in cytosol and also on the apicoplast membrane. So this suggested that there is overexpression of ATG8, which we further confirmed by Western blot. And but overall these results suggested that although ATG8 gets deconjugated from the membrane, but not completely. And why there is a higher level? So we checked if there is another enzyme that can process the ATG8 in absence of ATG4. So we looked into the literature and found a paper where they have nicely shown that this ovarian tuber unit cysteine protease can process ATG8 in malaria parasite. So next what we did, we checked the again like expression of this OTU in Plasmodium berghei and found that it expresses and localizes on the apicoplast membrane in Plasmodium blood stage parasites. So to study its function and having any overlapping function with the ATG4, we generated the knockout of OTU by using the similar double crossover homologous recombination and confirmed the knockout by checking its M-cherry expression, and then we checked its blood stage growth, where we found it grew normally and having no difference with the wild type parasite. And next we checked its transmission into the mosquito stages. So we checked the transmission of both knockouts, ATG4 and OTU, and we found that this ATG4 knockout, when and how we transmit it, we have infected mice because these are rodent parasites, and then we allow mosquitoes to probe for the blood meal. So when they are probing for the blood meal, they take the gametocytes along with the blood meal, and then their sexual cycle starts. So after 14 days, we checked its oocyst expression here, and we found a normal level of oocyst in wild type and knockout, and then we counted the oocyst number, which were also normal, and these oocysts form the sporozoites. So looking at this picture, there is a normal number of sporozoite production, that is called as sporogony, and then we counted these sporozoites that were also normal, that means comparable to the wild type parasites. And these midgut sporozoites travel to salivary glands and showing the similar load, that means there was no defect in transmission of parasite within the mosquito. So next we checked similar mosquito transmission for the OTU knockout parasite, and exactly it was like wild type parasites. So there was no defect in OTU knockout parasites. So we checked infectivity of both knockout sporozoites in the mice. So for this one, either we inoculated sporozoites by IV injection into the tail vein, or maybe we allowed infected mosquitoes to bite and inoculate sporozoites naturally in the mice. So in both cases, we found that OTU knockout parasites, the blue one, did not show much difference, like it was giving only 1 day delay in patency. But in case of ATG4 knockout parasites, we found that 50% of mice were not patent. That means after inoculation, they were not positive for the blood stage parasites. So and even if they came positive, there was a delay in pre-patent period. So this pre-patent period is when we inoculate sporozoites, the parasites appear in the blood. So that period, if we look at the wild type parasite, that takes 3 days to appear in the blood. But in knockout, you can see that there is 6 days or maybe 7 days. So there is a clearly 3 to 4 days delay. So next we checked its development in the liver. So like when we inoculated in the mice, sectioning mice and looking into the parasite is not easy. So we use the in vitro model where we infect HepG2 cells, that is a human hepatoma carcinoma cells, and after infection we harvested the parasite at different stages, where you can see that there is a normal development of both wild type and knockout parasites throughout its 40, 55 and 64 hour post infection. And after counting its number and also measuring the E of area, both were completely normal. And by looking at the maturation of the parasite, so by how we look, we stain with the merozoite protein antibody. So that is MSP1. So there is a normal maturation in case of OTU knockout, suggesting that OTU visually does not show any defect throughout its development process. So next we checked development of ATG4 knockout parasites, where first we inoculated mice and then we isolated the liver and we checked the parasite burden in the liver by measuring the 18S ribosomal RNA. So where you can see that at the initial level at 36 hours there is a normal level of parasites in both wild type and knockout. However, when we check that maturation stage, that is a 60-hour post-infection, okay, got it. So after 64-hour infection, okay, can you set it again, please? Screen. आया था वो again. चलो गया. So there is a significant decrease in parasite burden at 60-hour post-infection. So next we checked its development in the hepatocytes, and by observing up to 48 hours there is a normal development of the parasite. However, at 64 hours the parasite size is smaller and also there is no much nuclear division. So here you can see the EF numbers were normal, however the size was decrease of the parasite at 64 hours. So that means there is attenuation of parasites when there is no ATG4. So next we checked its development by staining with the merozoite surface protein, and you can see there is not much nuclear division and also the nuclei count is very less. And when we collected the parasite merosomes, so like when parasites mature they release the merozoites in the form of merosomes. So we collected and injected in the mice. So there was again decrease in the appearance of parasite in the blood. So suggesting that ATG4 is important for parasite maturation, and then it's a further transition from liver to blood. So next we checked like when in general parasites are attenuated in the liver, there is a defect in apicoplast biogenesis. That could be direct or maybe indirect. So because ATG8 is known to be conjugated on the apicoplast membrane, so first thing we checked development of apicoplast here. So we can see that there is a defect in apicoplast biogenesis. There is not much branching of the apicoplast here, but looking at other two organelles, endoplasmic reticulum and mitochondria, so there is some defect because we can see in the quantification. However, the defect is not like apicoplast. So that could be bystander defect, like general defect in maturation of the parasites, not maybe direct due to the conjugation and deconjugation of ATG8 on these organelles membrane. So next we checked like when sporozoites infect the hepatocytes. So these micronemes are exocytosed because they become superfluous there. So we checked its distribution, microneme distribution throughout the liver stage development, and we see there is no difference between wild type and knockout parasites, suggesting that ATG4 is not important, and it should not be important because it's terminal glycine exposed, and this process is only required for conjugation. So that's why we did not see any defect during this process. So next we checked its deconjugation in the liver stage parasites, and you can see that in case of wild type, like in the upper one, so there is a less ATG8 on the apicoplast membrane compared to the knockout one, showing that there is a deconjugation defect, and due to that deconjugation defect, parasite cannot mature properly in the hepatocytes. So then we try to generate double knockout, like deleting ATG4 and OTU together, but we failed to get that knockout. That means OTU partially compensate the loss of ATG4, but both are required for the parasite development. So that's why we could not delete it. So next we targeted ATG7, which is involved in both ATG8 and ATG12 conjugation system. So first thing we checked its expression and localization by fusing the gene with the HANM cherry. So in this transgenic parasite we found that it's mainly localized to the cytosol, and by fusing M cherry with this ATG7 it did not affect parasite development throughout the mosquito and mammalian host. So next we generated the ATG7 knockout. So first we tried to delete the gene directly, however we failed, suggesting ATG7 was possibly essential. So we took the conditional approach by using the FLP FRT based conditional mutagenesis system. So where we flirt the gene. So we flirt the VRF with 2 FRT sites, and then we transfect into a line that expresses the flippage. So that flippage does not express in the blood, it only expresses in the mosquito. So it keeps the gene function intact in the blood. So we get the silencing of the gene function only in the sporozoites, and then next we checked whether these modifications like putting FRT sites affect its development in the blood. So we checked the growth of the parasite and there was no difference. So our this engineering was completely normal. And then we checked the transmission of the parasite in the mosquito, which was again normal, and it should be normal up to at least sporozoite stage because gene is intact. But we get the knockout in the sporozoites that starts from the midgut, and we found normal midgut sporozoite development and also the salivary gland. And further these sporozoites also infected the hepatocytes, so suggesting that ATG7 is not important for mosquito stage development, and also further it's a transmission into the mammalian host. But when we checked its development further in the liver, so we inoculated the sporozoites in C57BL6 mice, and then first thing we checked its blood stage appearance. So we found that parasites lacking ATG7 failed to reach the blood stage, so suggesting gene is essential for its transition. So next we checked its parasite burden in the liver. For this one we harvest the liver after infection, then we do the real time PCR by measuring its 18S ribosomal RNA, which was less in case of knockout parasites, and also the MSP1 transcripts were also less. So they are like clearly indicating that there is a maturation defect when parasite lacks ATG7. So we next checked its development in the hepatocytes and found that like ATG4, so up to 40 hours there is a normal development. After that there is a extremely reduction in the size, and that we can here see by quantification, and also extreme decrease in the nuclei number. So parasites lacking ATG7 cannot divide their nuclei after 40 hours post infection. So and that here we counted the nuclei number, and also we again collected the merosomes and checked its infectivity. So any parasite that lacked ATG7 could not infect the liver to blood stages. And the next we checked its development like apicoplast development, where we found no branching. So there was absolute defect in apicoplast branching, and also there was defect in ER development. So organelles differentiation development affected in ATG7 knockout parasites. So this is showing the ATG8 localization in ATG7 knockout parasites, because ATG7 is a conjugating enzyme. So we should not see much ATG8 on the apicoplast membrane. So here we can see there is a separate signal, red and green. So when parasites like ATG7, there is no conjugation of ATG8 on the apicoplast membrane. So and again we checked, because this is involved in exocytosis of these rudimentary organelles. So for this one we checked the trap, that is the marker for micronemes. So here in initial level we found normal level of micronemes in wild type and knockout, but later they are not required, so they are exocytosed. In case of wild type parasites, in case of wild type parasites we see it's completely empty, the red signal is gone. However, that remains in the knockout parasite, suggesting that parasite lacking ATG7 failed to exocytose micronemes. So these important and essential functions led us to hypothesize this ATG7 as a drug target. So we targeted its ATP binding site, and for this one we generated its crystallic structure by using the Autotalk Vena, and then we compared binding of Mabry's library compounds by using Homo sapiens, yeast and Plasmodium falciparum ATG7. and we selected a score of 7.5, and having higher score more than like 7.5 for PF and for lower for human, like there could be no toxicity. So we found 100 compounds and then finally concluded to up to 15, and after the screening we found 2 active compounds. So those are named here BTB and RGF. So they inhibited plasmodium blood state development up to 2.5 micromolar. So next we checked its activity against the plasmodium. So this is showing the blood stage activity, where there is a like this is इसको सही करो बार बार तो पैरासाइट हाँ और आगे जाइए okay next bus. So parasite inhibited blood stage activity here against Plasmodium falciparum, and here you can see नहीं ऊपर वाला ही हम दबा रहे हैं. There's too much complication in this. हाँ जी हाँ जी. So there is a parasite development defect, and also that's happening due to lack of ATG8 ATG8 conjugation on the membrane here. So next we checked its activity against liver stages, and we found that RGF inhibited the liver stage but not the BTP that much. Probably there because we did that blood stage against Plasmodium falciparum and liver against Plasmodium berghei. So we again checked its binding, and we found that there is a one less hydrogen bond when ATG7 and these compounds binds. And these activities are specific to ATG7. For this one we express the recombinant protein and then set up the fluorimetric assay, and in presence of the compound we can see there is a decrease because it contains a aromatic amino acid. This is based on fluorimetry. So this is clearly indicating that these compounds inhibit specifically the ATG7. And to further improve the activity, we are modifying these 2 compounds by collaborating with my colleague Dr. Ramesh, and he has proposed modification around left and right hand side of these 2 compounds, and even we have improved the therapeutic or selectivity index of one of the compounds after modifying here, and the final conclusion is that ATG4 is important both in blood and liver. However, its function in the blood is compensated by OTU, but in case of ATG7, it's absolutely important for its conjugation and then finally maturation of the parasite in the liver. So we recently published this ATG4, and ATG7 is under review. And this is the group. I want to acknowledge all of them mentioned here for providing the reagent or contributing, and mainly this work was done by Akanksha and then contributed by Astha and Sabir. Thank you so much.
Thank you. So maybe one question already, we are getting late. Anybody have any question? You have anybody? Okay. No? Oh, yeah.
Mike,
Satish, very nice work. Starting from the identification of the target, characterizing them very well in its role in the parasite, and then leading to the drug development, which is a very good approach. How you are planning to improve on this selectivity as well as the efficacy of the molecules? Currently, that molecules you showed, they are working in micromolar range.
Yes.
Which is, I think, not good enough. So
not according to go and no go criteria. So we are modifying its left and right hand side by putting different aryl and heteroaryl groups. That is decided by the chemist, like how he can improve the activity. We have found very good, like its selectivity now, like its CC50 cytotoxicity is now greater than 500 micromolar. So that is good, but we haven't improved its activity. that is again like around 2 micromolar. So we are further modifying it, and let's see how we go ahead.
Okay.
Yeah, thank you.
All right. Thank you very much.
Okay.
So next talk is by Dr. Ferenc Petrozzi. Please come. Dr. Petrozzi is affiliated with the University of Debrecen in Hungary. He works in the area of organic chemistry, especially palladium catalyzed carbonylation, Suzuki-Miura cross-coupling reactions of IC substituted analogs. He is an active member of the talent management program. I think he also serves as a demonstrator for organic chemistry lab practices. So Dr. Petrozzi. Yeah, please go ahead.
Yes. Okay. Dear audience, let me introduce myself. My name is Ferenc Daniel Petrozzi. I came from Hungary, from Debrecen, from the Faculty of Pharmacy, from the Department of Pharmaceutical Chemistry. My research is about cannabinoids, and let me introduce the presentation about synthesis of fluorine-containing cannabinoid derivatives with various biological activities. Before I start my presentation, I would like to firstly say a big thank you for everyone for giving me a chance to present my work here. It's my first time in India, and we are having fun there. People are very kind and polite with us. Thank you very much. So I think most of us must have heard about weed, which is here. In scientific name, it's Cannabis sativa. It's a plant which contains many substances. I would like to highlight two main phytocannabinoids, the CBD and the CBG, which is cannabigerol and cannabidiol. They are non-psychotropic, so the psychotropic effect is given by the THC, the tetrahydrocannabinol, but these aren't psychotropic, so they cannot influence the consciousness like the THC does. They have several interactions in the human body, mainly the CB1 and the CB2 receptors, which are cannabinoid receptors, but they have also other molecular targets in the human body. A common property of these molecules is that they have beneficial pharmacological effect, such as antiviral, antibacterial, antioxidant, analgesic, anxiety, against anxiety, they can be used, and they have other undiscovered effects which are under investigation by several researchers. Only a few examples of systematic synthetic modifications in the scientific literature can be found about them. It's another common property. About cannabidiol on the left, it is allowed as a cosmetic ingredient in the European Union, but it has medical applications in different countries. If we check the Epidiolex and the Sativex, the Epidiolex contains 100 milligram per milliliter of CBD, and the Sativex is a mixture of CBD and THC in a 1-to-1 ratio. They can be applied as alternative medicines against muscle stiffness or spasticity or spasm in the indicated diseases. Cannabigerol doesn't have medical use yet, but it's used as a supplement, as CBG oil, for example. On this slide, I would like to highlight that about 20% of the commercial pharmaceuticals contain a fluorine atom. And if we check the previous year, the FDA approved 55 novel drugs, and 12 of them contained a fluorine atom, which fits the 20% of the statistics. The question can be arised why fluorine atoms are in the successful drug molecules. There are many articles which refers to them with several reasons. For example, fluorine is the second smallest atom after the hydrogen. So if we change the if you change the hydrogen to a fluorine atom, it won't really change or influence the property of the parent molecule. The fluorine and the carbon bond is one of the strongest bonds in organic chemistry, or the strongest bond which can be formed with a carbon and a fluor. So it gives the molecule a metabolic stability. The fluor is the most electronegative element in the periodic table, so it gives a very polarized bond which can influence the lipophilicity or the hydrophilicity of the molecules. And there are other reasons. For example, fluorine can participate in hydrogen bonds, or it can be a bioester of a hydroxyl group. Overall, these properties contribute to the ADME absorption, distribution, metabolism, and excretion mechanism, and they can influence these properties. This can be why, or it is discussed in many articles, why it is possible that many molecules which are biologically active contain fluorine atom. Nirmatrelvir was mentioned at the first presentation in that section, which is an antiviral medication. Perfluorohexyloctane is an eye drop for eye disease, and the fezolinetant to treat moderate to severe hot flashes caused by menopause. These were all accepted in 2023. And during this conference, I have seen many examples about drugs which contained fluorine atom. There are many fluorinated CBD and CBG derivatives which contain fluorine atom, and they have proved biological activity. For example, this first molecule is good for anxiety, antidepressant, and antipsychotic effects, and later it was proved that it has antinociceptive effects also. The following one can be potential against Alzheimer's disease, and the following can be used as treatment in cancer and in other diseases. I would like to highlight some more examples, two of the CBD, modified CBD derivatives. The first against autoimmune diseases, then the other in the article is described for a variety of disorders or diseases. And the last one is a CBG derivative, which is can be a potential a molecule for cannabigerol receptor ligand. The main difficulty is about them that most of the derivatives are patented, so which makes some obstacles to get information about the synthesis, the real properties, and this makes a bit hard to synthesize these molecules and get to the goal. In the next slide, I would like to talk about the Mannich reaction. This is the reaction what we really use during our synthesis. It's about a multi-component reaction. It contains three partners, a primary, a secondary amine or ammonia, which participates in the reaction with formaldehyde, and a carbonyl molecule, the red one, which contains an acidic proton next to the carbon atom, or the alpha carbon next to the carbonyl group. And it gives an amino alkylated product. There's another alternative which is called modified Mannich reaction. For example, for this with phenols, it can be occur the same amino alkylation procedure next to the hydroxy group of the phenolic ring. In case of primary amines, the first product can be produced, the second can be produced, an oxygen ring can be formed, and with other amines can be the simple amino alkylation, the first one produced. We have previous results in our research group. Different amines were tested, and in case of CBD, on the left, monosubstituted, I mean products with one oxygen ring were formed, and with different amines, in case of CBG, both mono-substituted and disubstituted products were formed. Okay, sorry. And an article has been accepted in the previous year. So our goals were to synthesize different fluorinated cannabinoid derivatives based on the previous experiences that the fluorine-containing molecules are potentially good pharmaceutical drug candidates, and we would like to improve the parent molecules' properties with the synthesis. And we have also heard during this conference in the section from the other side, the biological side, we have cooperations with different biologists and pharmacists who are investigating our substances, but our main goal is to synthesize them. We started with Mannich reaction, as I mentioned before, and we use 2,2,2,3-fluoroethylamine hydrochloride. It was in hydrochloride salt form. And on the first slide, we applied 2 equivalents of the amine, 4 equivalents of the formaldehyde. We completed this reaction in the dioxane. The product was formed, the disubstituted, which had 2 oxygen rings with 36% of yield. On the second line, we can see that we released the amine from its hydrochloric salt by adding sodium hydrogen carbonate to the amine, which was dissolved in dissolvent, in that case methanol. And when it was released, then the other compounds were added and the reaction had been started. In that case, interestingly, the monosubstituted product was formed with 1 oxygen ring. In the following steps, we use different amines. They contained 2 fluorine atoms. One was in form of hydrochloride salt, and one was a free amine base. On the first line, we used as a salt the amine component with the previous ratios of the reactants, and in dioxane we could isolate in very low yield the disubstituted molecule, which contained 2 oxygen rings. On the middle, the 2 reactions, I would like to highlight here that we tried 2 releasing methods. First, we used triethylamine as a base to release the amine from its salt, and at the second, as I mentioned before, we used sodium hydrogen carbonate. First, it was in dioxane, and the second in methanol. In the second case, we added more reagents, and as you can see, that in the second case, the 14/C conditions gave us a mixture of the mono and the disubstituted molecules, and the 14/A gave us only one product with low yield, the disubstituted molecule with 2 oxygen rings. And finally, we tried the free base, which gave us a monosubstituted product with a very good yield. In case of CBD, our final examination with these kind of amines was the 2-fluoroethylamine hydrochloride salt. We tried to release the amine with the previous condition with sodium hydrogen carbonate. We applied these ratios, 3+2 equivalents of the amine, 4+4 equivalents of the formaldehyde, and 3+2 equivalents of the sodium hydrogen carbonate. We used methanol, and the reaction time took 2 days. And in that case, the monosubstituted molecule was formed with good yield. Then we followed our investigation of harmonic reaction of the cannabinoids with CBG and the 2,2,2,3-fluoroethylamine hydrochloride. At the first case, we used only the hydrochloric salt. As I mentioned before, it's similar to the CBD version. We could isolate with the dust-substituted molecule, and when we applied releasing the amine from its hydrochloric salt, we could isolate the monosubstituted molecule. It was followed by the free amine, which contained 2 fluorine atoms, and we could isolate the monosubstituted amine in that case with a very good yield. And finally, in case of CBG with 2-fluoroethylamine hydrochloride, we used first as a salt and for the second also, but the main difference was the solvent. In case of methanol and dioxane, there were no significant difference between those conditions, because both gave us a mixture of the monosubstituted and the disubstituted molecules. As you can see on the first line, the main component was the monosubstituted. At the second, the ratio was close to 1:1. And then, sorry. We were about to try aromatic amines. So this is why we used aniline derivatives, which contained 3 on the third position, on the fourth position, a fluorine atom. Starting the work with CBD, we used the same conditions, I mean the same ratios in case of CBD and CBG. I mean 3 equivalents of amine, 3 equivalents of formaldehyde. We proceeded the reactions in methanol, used solvent, and we applied reflux. These aromatic anilines gave us very good yields. In that case, monosubstituted product was formed with very good yield. It was followed by the para-substituted version of this aniline. In that case, also the monosubstituted derivative was formed. And in case of CBG, in this case, we can see that the monosubstituted derivative was formed with 1 oxygen ring. But in case of the para-substituted derivative of this amine, we could isolate a mixture of the mono and the disubstituted CBG derivatives. And as we can see, in excess of in the mixture, the monosubstituted derivative was. Firstly, from the substituted CBD and CBG derivatives, the anti-SARS-CoV-2 activity was examined by collaborative partners, except all of the CBD and CBG derivatives were investigated except the aromatic ones. They were investigated on Vero E6 green monkey kids cells by partners. So at the introduction, I mentioned that CBD and CBG have antiviral activity. In that slide, we can prove with the results that yes, it's true, because CBD and CBG have antiviral activity. But I would like to mention also that in the concentration when they are active, it's a very cytotoxic concentration. Our derivatives had moderate antiviral activity also, but first I would like to highlight it here that we have found a structure relationship and structure-activity relationship that the substituted molecules didn't really have cytotoxic effect, and they have also antiviral effect. The investigations were followed by the antibacterial activity discovery, also with partners. As I mentioned before, it's true that CBD and CBG had antibacterial activities on gram-positive strains, bacterial strains, and unluckily our derivatives didn't really have as good antibacterial effect as our parent molecules. Then CBD and CBG molecules were investigated by partners on human renal cancer cell carcinomas. I would like to mention here that both CBD and our molecules had almost the same effect on these cells. There were some promising molecules which can be important to examine there in other assays. And in case of CBG, it can be also said that there are some molecules which should be examined, but they are some of them were similar to the parent molecule. And in that case, I would like to mention also that based on the previous results in antiproliferative assays, it can be said, and we wish that there will be selective effect on selective antiproliferative effect. I mean that the cancer cells so our molecules will be active on cancer cells, but not active on healthy cells. So as a summary, I would like to mention that we have successfully synthesized fluorinated cannabinoid derivatives with Mannich type reaction. The anti-SARS-CoV-2 activity and the antibacterial activities were discovered or were investigated by partners. We can say that these compounds had weak antibacterial and moderate antiviral activity. The other aromatic, the other CBD and CBG compounds which were modified with the aromatic amines, we would like to send them and examine them because we don't have about them results yet. The antiproliferative effect of some fluorinated cannabinoid derivatives were investigated. Most of the synthesized derivatives exerted an effect similar to CBD and CBG, but some of them were promising, so we would like to do more assays. And the examination of the effect on skin cells, antioxidant assays, and antiproliferative tests on other types of cells are under process now. I would like to highlight here my supporters, my supervisor, my colleagues who gave us very much help, and I'm very grateful for them. I would like to thank for the different scholarships and applications and my collaborating partners who investigated our molecules. And I would like to thank you for your kind attention.
Thank you. So, if anybody has any question.
Sir, well, your presentation is really wonderful. I really enjoyed all the facts. But the question I have right now is that you have mentioned different derivatives of CAB and CAG. My question is, sir, have you ever, put attention on the side effects of these particular derivatives? Like, there are side effects of this medicine if we, consume it for the market or the people who are really in need?
Yes, we didn't really investigate the side effects. Our main focus is on the effects, biological activities, if they have any or not. Later, if they are promising, or if we found a promising molecule, it would be, of course, examined the side effects. But the first goal now is to get to that point that when we have some samples on the flask which are promising, and then we will extend the examination.
All right, thank you, sir.
All right. Thank you very much.
Okay, thank you.
We still have 2 more talks. One more talk has been added, so I will request people to bear with it and stay for 2 more talks. One we are going to have now, and after that, 1 more. So the next talk is by Dr. Richard Kazar. Dr. Richard is also from University of Debrecen, Hungary, and in the Faculty of Pharmacy, Department of Pharmacology. His research centered around the pharmacological characterization of various CBD derivatives. So I guess you heard the CBD derivatives and more from Dr. Zahar. One more talk after this.
Previous point. Thank you.
Request the speakers to reduce the time to 5 minutes. 5 minutes.
It's okay. No, it's okay. So, welcome everybody, dear audience. And first I want to thank you to everyone invite me here in this great conference. It is an honor to present in a place such as this. This is wonderful. But after that, let's jump into it. Thank you for the introduction. So, do not waste the time. First, I have to talk about my main research about cannabidiol, aka CBD. So, what is this? My colleague before just introduced it pretty well, so I will just say a few words about it that we should know, as it's a phytocannabinoid. In the latest years, researchers pretty focusing on it. Of course, it have the famous cousin THC. The main difference between these two phytocannabinoids that CBD does not have psychotropic effect, rather the opposite. It can prevent the psychotropic effect of the THC or can modify it a little bit. So, but not to talk about this,
there are some different and really interesting pharmacological effects of the molecule. So I will talk about not all of them, of course, because as a researcher I can just focus on a really limited part of the whole pharmacological profile. But as you can see, it have many effects, and they are proved, most of them. So I will talk most briefly about antioxidant effects because it's closely related to my research. In our research group in Debrecen, we are mainly focusing on cardiovascular diseases since it is the most prevalent disease around the world, and it causes most of the deaths on Earth among humans. So, antioxidant properties. We should start with indirect antioxidant properties. CBD, it is proven that after administration it can improve the superoxide dismutase protein expression in the cells. This enzyme is responsible for hydrolyzing the superoxide radicals, in which with this method it can prevent the cells from oxidative damage caused by free radicals. It also can increase the effect of glutathione peroxidase, and with this case the glutathione levels are rising in the cells, so they can this action can protect the cells with a lot of glutathione against the oxidative damage. Apart from the indirect antioxidant effect, there is some direct scavenging ability of the molecule. As you can see, there is an aromatic ring and two phenolic groups. These delocalized electron structure is mainly the responsible for the direct scavenging ability. And yeah, there are many other effects, anti-inflammatory effects. Just a few words. It is proved that CBD is an agonist on PPR gamma receptors, and there are several researches where they found that other PPR gamma agonists, such as rosiglitazone and others, can pretty helpful in Alzheimer's disease, but of course in connection with CBD it is needed far more investigation. And yeah, as my colleague mentioned before, the main two receptors which is deals with cannabinoids, endocannabinoids and everything in the human body is CB1 and CB2 receptors. The most interesting thing about this molecule, CBD is an antagonist on these receptors. The opposite of THC. THC is an agonist on CB1 receptor in the brain. This is the main cause of the psychotropic effect. So this is why it can modify it. And this is one of the reasons it have anxiolytic effect. The other is because it's an agonist on the serotonin 1A receptors. But this is another story. Here you can see with red the research, what have I done more briefly, and with blue I just highlighted those that we want to investigate in the future. So as you could see in the previous colleague, the pharmaceutical chemistry, the Department of Pharmaceutical Chemistry synthesized modified CBD derivatives. I will refer them as CBD1, 2, and 3. So they are modified molecules in the aromatic ring. Since there is limited information about modified CBD molecules, the natural CBD and the other phytocannabinoids are researched, investigated briefly, but there is really a lack of information about synthetically modified ones. Our main focus is, as I mentioned, our cardioprotective effect and antioxidant mechanisms. As you saw before, they are synthesized with the Mannich reaction. I'm not the expert of field. If you have paid attention, you could see how it works. And our main aim was to check it if these molecules are valuable in the cardiovascular system, since it's a pretty common disease among everywhere. Now I have to mention some of my methods and materials. First, I want to say some words about the cell line, what we worked with. It is the H9C2 cell line. These are cardiomyocytes, rat cardiomyocytes, and they are immortalized, so they preserved the proliferation ability. So that's why we can work with them. First, we checked the bioavailability, or rather the toxicity of the cells. If can we apply it or not? Are they toxic? So we determined the IC50 values with an MTT assay. After that, we observed the antioxidant properties with the Turlock's equivalent antioxidant assay. And then we tested the molecules in several conditions, which includes oxidative stress, hypoxia. We challenged the cells with hydrogen peroxide. We made a trypan blue exclusion also, and then we just investigated the indirect antioxidant proteins with a Western blot analysis. So my first slide of results shows the cell viability graphs. As you can see, the untreated cells have 100% viability, and we can say that in the concentration of 1, 3, 10, and 30 micromole of the new compounds, and also the CBD, they does not have any toxic effect. And maybe it is good to say that the new compounds are equally not toxic, or they are slightly less toxic than the CBD. So if it is needed, we can apply more of them than the parent molecule, because their toxicity is a little bit lower. But moving further, as I mentioned, we challenged the cells with hydrogen peroxide just to stimulate some oxidative stress. We investigated that in the concentration of 125 micromole per liter, the cell line just got a really big damage. The cell viability just decreases from 100% until 70, as on every graph you see the second column. And we was curious if we apply these compounds in the non-toxic concentrations, can they elevate the cell viability? And as you can see, slightly in 1, 3, and 10 micromole, all of the compounds was capable for this. And then we made the trypan blue exclusion. In this experiment, we made a 4-hour simulated hypoxia for the cells in a hypoxic chamber, where the oxygen level is decreased to 1%, and of course with the new media they haven't got any glucose. So this can stimulate or simulate myocardial infarction in cell level. Of course, it's not a myocardial infarction, but it's really close to it. As you can see, in the normoxic group, there is about 86% of viability. and there is not really a big difference between the treated and the control group. But in hypoxia, as you see, the control group who is not treated with anyone, after 4 hours of hypoxia and 3 hours of reoxygenation, the control cell line have 60% viability. And the compound could elevate this up to 87% by the second molecule. So they can protect the cells from this harm, and it's pretty nice. Just to further prove this, we made an LDH releasing assay with the same setup, 4 hour hypoxia and 3 hour reoxygenation, and we saw that from the new molecules, the labeled 1 and 2 in 3 and 10 micromoles concentration, they could greatly reduce the cytotoxicity, which was 30% in the control group. And then we wondered about the direct antioxidant effects. We made an ABTS assay or Trolox equivalent assay. It is the same. So it is we make an artificial free radical which have the color green, and rather if they more potent, the decolorizations ratio will be bigger. So it is as simple. And the final conclusion of this slide, we can say that the new compounds have a moderate activity compared to CBD, but they are still comparable except the third molecule. And finally, we tested these compounds in connection with the indirect antioxidant capacity with protein catalase, superoxide dismutase, and homoxygenase 1. Those are dedicated, they have a pretty much effect in hypoxia or oxidative stress related conditions. As you know, catalase is the natural enzyme which is responsible for hydrolyzing hydrogen peroxide in living cells. And we saw that in hypoxia and also in normoxia, they could greatly elevate the levels of this enzyme, which is great. So maybe this can be the background of the protective effect of the second molecule. As you can see in hypoxia and also in normoxia, it could significantly elevate the levels of these enzymes. After that talk about the superoxide dismutase, we also saw a slight elevation, but it wasn't as outdating or outstanding like in the case of catalase. And finally, in homoxygenase 1, we also experienced that the new molecules could really elevate the levels of homoxygenase 1, which is closely related to autophagy and other protective effects in the heart. So, and in this part, they are just beating the mother compound, the CBD. So the final conclusion of this experiments, this research, that the new compounds have a comparable antioxidant effect, and in some cases, as we saw the last slide, the last part, they are even better than the original molecule. And they have a great cardioprotective effect, of course, in the cell level. So that is why we will need further investigation. The most potent molecules, we will perform some ex vivo experiments on isolated rat hearts And we want to check the direct antioxidant profile more briefly with other tests. But before I finish, I have to say some words about my colleagues' work with the fluorine containing CBD molecules. We have already started this new research with 6 of his compounds, and I just want to show you in 2 minutes the results. These are previous results, so we need to some further information about them, just to confirm they are true or not. Here you can see the logarithm IC50 values of the molecules on the cell line H9C2. And the message is the same. They are slightly less toxic than the CBD. The other, the ferric reduction antioxidant power antioxidant assays results, we can say the same, that they have comparable antioxidant activity as the mother molecule. From the new compounds, the labeled 17 is the most potent for the first C. And after that, with the oxygen radical absorption capacity assay, we can confirm that, or even highlight that the compound 17 have an outstanding activity in this assay. Of course, we have to make some repetitions and confirmations, but it seems pretty promising. And after that, the ABTS assay, which is performed with the previous 3 molecules also, we can say also that they have slightly moderate effect, but still comparable to the mother molecule. And as I'm reaching the end of my presentation, I want to thank to all of my colleagues from both departments, from the Department of Pharmacology and of course from the Department of Pharmaceutical Chemistry, because without them I do not have any compounds to test. So yeah, and after that I want to thank to anybody who is made this research capable, and of course the foundings that make the financial background. And for you, thank you for your kind attention, and if anyone have a question, I'm here to answer.
Thank you. So, anybody? Anyone? Okay, I will ask a small question. Do you have a plan to these have got multiple effects, cardioprotective, then fluorine derivative, antimicrobial and things like that. Do you have a plan to study the mechanism? How does it increase the, the glutathione, SOD and peroxidase, glutathione, all those things? Does it bind to the promoter region, or it induces something which does?
Yeah, thank you for the question. Yeah, we have a plan about this. As superoxide dismutase, we have already tested that they are capable to increase the expression, but the reason how they can, it is the more interesting. I had no plan, maybe my supervisor had, but he haven't mentioned
I will tell your supervisor to force you.
I will definitely write this idea down and ask him that what could be the reason why. That's a good idea. Thank you.
Thank you. Thank you very much. So the last talk of
asked to go and sit inside the room, rest on a bed, and that's it. So it's time that we look at the anti-resorptives. But when I look at the anti-resorptives, then they will again change the balance. So well, nice to look at better therapies, more precise therapy. So this is where we looked at, I've thought of this part, that there should be a gene which is responsible for this. We looked at the want signaling pathway. It is a non-canonical pathway which can look after many, many things. But in this, we looked at the beta-catechin, looked at the SOST gene specifically. Why did I want to look at SOST gene? Because sclerostin protein, which is secreted there, is the only protein which is the crosstalk between bone forming cells and the bone killing cells. Well, looking at that, how do I do it? I must look at the siRNA, which is going to actually just precisely do what I want. Well, good enough. Then, how do I do it? I want an siRNA, so we decided the siRNA, which is going to be SOST. How do I deliver it to the place? So I looked at conventional therapy and nanotherapy. So we looked at nanoparticles. When we started looking at nanoparticles, we found that there could be immense possibilities. Looking at so many nanoparticles, we decided, okay, gold, which is most non-immunogenic, and chitosan, which can help us to reach the target site where we wanted. So after this was the design of our experiment. We made the gold chitosan nanoparticles. We optimized, characterized, functionalized them for in vitro as well as in vivo. In vitro is when I want to look at the cells, and in vivo is when I want to send it into the animal for a clinical translation. Then we had to look at all the, when I showed you the 5 steps, it just shows that the bone has to undergo differentiation from the embryonic stage to pre-osteoblast to osteoblast, and then it differentiates into osteoblasts and it goes into mineralization, which tells us that, okay, we have reached a point. And then we do the gene silencing and the oxidative stress to see whether we are reaching. In vivo also we have done, but I'm not going to show today. So we started the optimization with the Box-Beckenham design, where we had 2 constraints, and when we looked at the size and everything, we came upon very beautiful chitosan reduced gold nanoparticles. We did the characterization, and this is the scheme where we've shown that we have done a layer by layer constructing or synthesizing of the gold chitosan nanoparticles. We have to see the size, we have to see the charge. Why I don't need the charge? Because a negatively charged siRNA molecule has to go into a positively charged polymer chain, otherwise we won't be able to encapsulate it or stabilize it as we would like it to be stabilized. Then we looked at the other morphology, because you have to see it through the electron microscopy, whether it is transmission or scanning, which will give you the size, the shape, and we do the DLS, which we had done the DLS here to get the size. So we get beautiful nanoparticles, but since we have gold, we also must do the EDX to know whether the gold is actually there or what we are looking at is just an artifact. We then did the elemental analysis also to understand that our characterization and the cells, the nanoparticles which we had made were accurate, so that now we will worry about functionalization. How do I functionalize them? I must stabilize it with the help of the PEI, that is polyethylene imine. Why do I need that? Because I would require it for transfection, and it will facilitate its entry into the cells. So we did the binding assays to see whether the binding was correct, and then we hypothesized that it would be internalized like this. After that, next, please. Yeah, okay. So this was the entire mechanism which we hypothesized, so that it would be internalized, stall the beta-catenin pathway, and it would also help us to see the osteogenic markers run next to an ALP. That would tell us whether we have been able to, had it been, no, nahin, abhi. सब कुछ चला गया, आ गया. Okay, so अभी आगे नहीं करना प्लीज. So this is what आगे नहीं करना प्लीज. मैं कुछ नहीं कर रही हूँ. Okay, okay. So what I had explained was that the want signaling, which is a frizzled pathway, non-canonical, which sits on the surface, if the SOST gene expresses, we will have the osteogenic markers. If it silences, we will have reduced sclerostin, because we have seen enhanced sclerostin in people suffering from osteoporosis. And this is how we will take the cells, make them into pre-osteoblasts and osteoblasts, and then check it out. So we started with two sets of cells. One is the bone cells, which are the MC3T3 cells, and the other is the mouse embryonic fibroblasts. So both of them were differentiated to here. We did the internalization to see whether we had done the binding, and when we had done the binding with PEI, were we able to internalize? Yes, we could see you can see the green fluorescence, which shows that it was internalized. We had to see the cellular uptake prior to understanding whether the gene knockdown experiments will take place. Here, I'm showing you together on MC3T cells on the left side and mouse embryonic fibroblasts on the right side. The upper panel where you can see the blue, that is nothing but alkaline phosphatase, and the red one is the alizarin red. Alizarin red tells us the mineralization of the cells, and the alkaline phosphatase tells us, the blue color shows that the cells are differentiating. That means my osteoblasts have to become mature, then only we will see the sclerostin protein. So we had to optimize which day, from 0 day to 28 days, which day am I going to get the highest amount of sclerostin. It was from day 7 to day 14, we got the almost 20 folds higher amounts of sclerostin. After 14 days, post 14 days, means till 28 days, it declined. So this is what we are showing, that mineralization starts increasing. That means my extracellular matrix has more calcium deposition, hence the color becomes more and more red. So once we understood that this is happening, seeing is beautiful, but we also have to see whether the gene expresses. So we had to see the osteogenic markers, whether RUNX2, ALP are also enhanced after mineralization or not. So yes, the lower side, you can see that panel, the graphs clearly show upregulation of the osteogenic markers. So we moved on to see, now that we had already prepared the siRNA nanoparticles, whether they showed a knockdown, and if you see very carefully, it has completely knocked off the thing. If you see the panel here on the right side, the third graph, you can see that it has completely silenced it. So it was very clear that the results were efficient delivery into the system, efficient silencing of the SOST gene. Well, once we did this, we had to see whether there was oxidative stress which was being formed, and oxidative stress, very happily, my previous speaker gave a very good brief, where we are looking at the GSH and GST. Glutathione family is very important, detoxifying family. We are also going to look at the SOD, because two things are important. The moment something goes into the cell undergoes an oxidative stress. What do I mean by that? I mean that it is going to generate so much of the reactive species that it might land up damaging the cell membrane. So first we look at the lactate dehydrogenase, so that we understand whether the integrity of the membrane has been damaged. If not, then we want to look at how will it do, whether it has increased the glutathione family or it is looking at the nitric oxide. So the moment I look at the ROS, I must simultaneously look at the reactive nitrogen species also. So we look at both of them and see whether lipid peroxidation is happening, whether it is a nitrosative stress, whether it is an oxidative stress, or it is a combined nitro-oxidative stress which has forced the cells to, which has become an outcome of that, because nitric oxide is a double-edged sword. If I allow too much of NO, my blood vessels will dilate, it will create a problem. If I have less of NO, I am going to kill the cell anyways. So looking at the SOD levels, looking at the entire glutathione family, the GSH, GSSG, and glutathione reductase and peroxidase. Looking at all of them, we see enhanced levels of SOD. That means our SOST gene has been silenced. So putting the entire story together, I'd like to summarize that we actually have created a wonderful strategy by way of which we are only silencing the SOST, which can comfortably allow the bone formation in the system so that the homeostasis becomes normal and the bone forming cells become equal to the bone eating cells. So, and we have seen this in both of them. That means we can differentiate the fibroblasts as well as the osteoblasts, the pre-osteoblasts, which we have already taken. So this has already been accepted by nanomedicine. By Monday next, this will already come into the net, and you people can start looking at this paper. The outcomes are beautiful. We have a cost-effective delivery system, high impact for human health, completely ready for translation. Patent has already been applied, publication has come. The research which has been established also, it is very important as the SDG goal of, because it is of global importance. We cannot deny osteoporosis anymore. People have to go and start looking at all the patients who have osteoporosis. Well, all the work my students have done beautiful, because I started with Vijay, who has moved on as a Marie Curie Fellow. Largi has moved on with the Newton Bhabha Fellowship. The same work is now being followed by Mansi, who is also moved on to do an miRNA, and she has got the Commonwealth Split Site Fellowship for this year, which she has gone to UK to currently avail. And this is my tree, which is growing, ever growing, a list of 22 students. I don't know if any more will be added, but I'm already towards the end of my career. And thank you very much. The last slide of acknowledgement, I completely acknowledge DBT for this grant, and thank you very much. And I'm sorry I have kept you away from the lunch, but I tried to be as brief as possible. Any questions?
Anybody have?
Yes, yes.
Yes, one question. Anybody has? Not working for any, I can speak now.
Sir,
No question.
no. Thank you. Yes.
Even I have stage 1 osteoporosis.
Okay, all right.
So, but the technology these days, people are recommending, even these ortho, that the experts are recommending, they call it PRP, some PRP therapy or something.
Yes, PARP therapy, yes.
PRP.
Right.
How that...
No, that I showed when I showed that there are already anti-absorptive drugs and monoclonal antibodies. So they are biologics. And these biologics, your PARP is one of them. Like I just mentioned, after all, even if we put a monoclonal antibody, they are going to induce gene expression of something, and they are also going to see the oxidative stress. Now, what are they doing? If I do INOS, it will take the immune pathway. If I do NOS, then I go into this bone pathway. So I have to be very specific in checking the subset of NO molecule which is going to be forwarded. And monoclonal antibodies also have a lot of side effects because they are not specific. Although we say they are highly specific, they go and bind, but the secondary and the tertiary stage is not under control. Hence, we wanted to do this entire mechanistic studies. For you also, if you do the PARP, it is good to have a checkup of your oxidative stress levels in your sera, which will help you to decide how to revert back. The main thing is to keep the bone homeostasis normal. That's it.
All right. Thank you, Dr. Anita. With this, I thank all the speakers, although they are not here, and especially to all of you for staying until this time. So with this, this session is closed, and yes, want to say something?
Thank you, ma'am. Now I want to just welcome Dr. Maya Sharma to felicitate Professor Anita Kamra Kirori, Mal College, University of Delhi. Thank you everyone to enlighten us with this informative sessions. I hope it will be beneficial for all the participants and academy persons and research persons and guests. Now I would like to request everyone to join for the lunch and poster session, and after that we will again gather here for the next session.
सर ये हमने शुरू ही काम इसलिए किया कि ओस्टियोपोरोसिस जैसी